All right, I want you to picture our planet, but wind the clock way back. Over 500 million years ago, the Earth's oceans are vast, they're mysterious, and they are poised right on the edge of arguably the greatest biological revolution in the history of our planet. Welcome to This Explainer. Today, we're diving deep into the incredible evolutionary blueprints that actually gave rise to modern aquatic invertebrates. Because within a remarkably short window of time, an event we call the Cambrian explosion, simple organisms rapidly evolved into complex creatures. We're talking distinct tissues, intricate organs, powerful muscles, and entirely new, active ways of surviving. It's wild to think about, but every crab scuttling across the sand, every octopus navigating a reef, even us, frankly, we all share architectural innovations that were first drafted in those ancient seas. Okay, let's dive into this roadmap for today. We're going to start by exploring the Cambrian blueprint, then trace how life builds complexity through levels of organization. From there, we'll look at the physical architecture of symmetry and cephalization before zooming way in on early development with embryology and germ layers. Then, we'll unpack the major evolutionary split between protostomes and deuterostomes and wrap it all up with the evolution of body cavities. So, Section 1, the Cambrian blueprint. Let's take a look at invertebrate phylogeny. This period was absolutely crucial. It was the definitive era when the essential architectural rules for modern invertebrates were laid down. And this wasn't just some slow, gradual shift, It was a profound, massive transformation in how biological life operated underwater. During the Cambrian, we see the very first appearances of distinct tissues and internal organs. We start seeing standardized patterns of early embryo development. We get body symmetry, the incredible innovation of cephalization, which is basically the evolution of a distinct head, and the rise of segmentation, plus the formation of 3 germ layers and a vital internal cavity called the coelom. These specific evolutionary leaps sparked a massive predator-prey arms race. They literally laid the absolute groundwork for the mind-blowing diversity of invertebrate life that we study today. Moving on to Section 2, levels of organization, going from cells to systems. Now, to really understand these modern organisms, we have to trace how biological life built itself up, going from simple, isolated structures into unified, complex systems. It's like a scaffolding of biological complexity. We start at the most basic baseline, the protoplasmic level. Then we move up to cellular organization, which is where you find creatures like porifera, the sea sponges. Step 3 is the cell tissue level. Think of cnidaria here, like jellyfish. Then, the complexity scales up again at step 4 to the tissue organ level, beautifully represented by platyhelminthes, or flatworms. And finally, we hit the pinnacle of invertebrate complexity, the organ system level. This is where highly active organisms like arthropods and mollusks live. It's an amazing evolutionary ladder of increasing specialization. All right, Section 3, Symmetry and Cephalization. We're looking at body plan geometries. Once cells actually organize themselves into tissues and organs, they have to physically arrange themselves in 3-dimensional space to form a functional body. And this physical shape completely dictates an organism's lifestyle. We generally classify these body plans into 3 main buckets. First, you've got asymmetrical bodies, like our sponges, which basically have no distinct shape at all. Next is radial symmetry, which we see in coelenterates, like sea anemones. These guys have a top and a bottom, usually a feeding and non-feeding side, and they're typically sessile or planktonic. That means they just sit still attached to something, or they drift. Finally, we have bilateral symmetry. And wow, this is a massive evolutionary leap. Seen in annelids, arthropods, and mollusks, bilateral animals have mirror image left and right sides. This specific geometry is the game changer that enables active, forward-directed, coordinated movement through the water. And tied directly to that forward movement is cephalization. This is the presence of a distinct head that bears sensory organs and central nervous tissues right at the interior or front end of the animal. Think about it. If you're a bilateral animal actively moving forward through your environment, it makes complete evolutionary sense to pack all your sensory equipment right at the front, where you're entering new space. This brilliant adaptation allows invertebrates to perceive and react to their world in highly sophisticated ways, navigating everything from ancient oceans to modern coral reefs with pinpoint precision. Let's move to Section 4, embryology and germ layers, the stages of early development. To truly appreciate how a complex adult body takes shape, we actually have to go right back to the very beginning of the life cycle. Early embryogenesis is what ultimately dictates the final invertebrate body plan. It all starts when the developing organism undergoes cleavage, basically rapidly dividing its cells to form a hollow sphere called a blastula. This little sphere features an outer layer called the blastoderm and a fluid-filled internal cavity known as the blastocoel. This initial, seemingly simple hollow sphere is incredibly important because it sets the stage for the crucial next step in development, a process called gastrulation. That is where the true complexity really begins to unfold. And this gastrulation process brilliantly illustrates the two main frameworks of germ layers that construct metazoan bodies. So think of germ layers as the primary building blocks of the animal. On one hand, we have diploblastic organisms. They develop two true cell layers. You get an outer ectoderm that becomes the epidermis, and an inner endoderm that lines the gut. In between them is this non-cellular jelly called mesoglea. But on the other hand, we have triploblastic organisms. These creatures introduce a completely revolutionary third layer, the mesoderm. This central layer sits right between the ectoderm and endoderm, and it is entirely responsible for generating complex muscles and major internal organs. It's a huge deal.
Section 5. Protostomes versus Deuterostomes, tracing developmental patterns.
Okay, so once an animal is established as a bilateral, triploblastic organism, a massive evolutionary fork in the road happens during its early development. When that early embryonic cluster of cells folds inward, it creates an opening, and we call that a blastopore. The fundamental split in invertebrate evolution depends entirely on what that opening ultimately becomes. In protostomes, which literally translates from Greek to mouth first, that initial opening develops into the animal's mouth. But in deuterostomes, which means mouth second, that initial blastopore forms the anus first, and the mouth forms completely separately later on the opposite end. It is a stunning embryonic divergence that literally splits the bilateral animal kingdom right down the middle. And finally, Section 6, Evolution of Body Cavities, exploring the coelom. Now that we have a grip on developmental pathways, let's explore their internal plumbing. The major evolutionary breakthrough here is the coelom. By definition, a true coelom is a fluid-filled space situated right between the digestive tract and the outer body wall. So why is this so important? Well, the mechanical advantages are just staggering. First, it acts as an internal shock absorber to cushion all those newly evolved suspended organs. Second, it functions as a hydrostatic skeleton, giving soft-bodied creatures the pressure they need to move with incredible force. And perhaps most importantly of all, it gives internal organs, like a beating heart or digesting gut, the physical space to move, function, and grow completely independently of the outer body wall. Visualizing how this cavity actually sits inside the animal is crucial to understanding how they work. We can basically map exactly where that mesoderm interacts with the gut. In the acoelomate plan, like we see in a flatworm, the mesoderm completely fills the space. There is literally no cavity at all. Then we have the pseudocoelomate plan, like in a roundworm. Here, there is a cavity, but it's only partially lined by mesoderm tissue. And finally, we see the true coelomate plan, like in an annelid. Here, the mesoderm completely splits to create a perfectly lined, dedicated, fluid-filled cavity. This right here is the anatomical gold standard for advanced mobility and complex organ development. Summarizing these evolutionary endpoints really shows us where modern groups land today. Flatworms are forever bound to that simpler acoelomate structure, and roundworms are tied to the pseudocoelomate plan. But the highly complex groups, mollusks, arthropods, echinoderms, they fall entirely into the true coleomate category. Developing a true coelom was essentially the biological key that unlocked a whole new world of evolutionary possibilities. Which brings us to this final, really profound thought to chew on. How do these ancient Cambrian blueprints continue to dictate the biology, the movement, and the evolutionary potential of modern life in our oceans today? Because every single time you see an invertebrate swimming or crawling through the water, you are watching over 500 million years of architectural innovation actively at work. From the exact timing of their cell division as embryos, to the bilateral symmetry of their bodies, right down to the layout of their internal cavities, the fundamental rules of their lives were written in deep, deep time. I really hope this explainer leaves you eager to look at a simple sea star, a jellyfish, or a crab with a completely new, much deeper perspective. Thanks for learning with us today.